Okay, I think we'll get started. Okay, so I'd like to welcome everybody to the SEBS virtual learning experience, Microbiomes and the Effect of Urbanization, with Dr. Maria Gloria Dominguez-Bello. My name is Brian McGonigal. I am the Manager of Alumni Community Engagement for the School of Environmental and Biological Sciences, as well as the New Jersey Agricultural Experiment Station. Just a couple of notes before we get started. Our event will be recorded and posted online for subsequent viewing. If you have any questions, please feel free to enter those into the question and answer box, which is at the bottom of your Zoom screen. If you hover your mouse over, you'll see the Q&A box. Please submit your questions there. You can submit your questions at any time during the presentation. You don't have to wait until the very end. So I'd like to talk a little bit about our speaker. Dr. Maria Gloria Dominguez-Bello is a Venezuelan-born American researcher at Rutgers University. She did her bachelor's degree in ecology at Universidad Simón Bolívar in Venezuela, and her MSc and PhD at Aberdeen University in Scotland under a British Council scholarship. She returned to the Venezuelan Institute of Scientific Research in 1990 for 12 years until 2002, then moved to the University of Puerto Rico, where she was a professor for 11 years until 2013. She then moved to NYU for 5 years, and has now been at Rutgers since 2018. She is currently the Henry Rutgers Professor of Microbiome and Health and the Director of the New Jersey Institute for Food, Nutrition, and Health, or IFNH. She is a Fellow at the American Academy of Microbiology and of the Infectious Disease Society of America, and belongs to editorial boards of several scientific journals. Her current lab studies the impacts of modern practices on the microbiome and strategies for restoration, working on gradients of urbanization in South America through global synergistic collaborations. Her current interest on lifestyles provides a perspective of diversity to the microbiota across diverse populations. She is leading the global initiative of the Microbiota Vault to preserve the diversity of microbes relevant to human health. So without further ado, I would like to present Dr. Dominguez-Bello.
Thank you, Brian, and thank you, Mary, for inviting me to share our work. Good afternoon, everybody. I'm trying to share. I hope you can see the entire screen now. Yes, Brian, can you confirm?
Yes, we can see it.
Great. So I'm going to share with you our work, the work we do on microbes and how changes in lifestyle change our microbes and why this is important. And so let me start with just reminding us that sanitation and medicine have been extremely important. Their highly antimicrobial activity has been crucial to control infectious diseases. So this has allowed us to really live in big groups, big cities, and controlling epidemics. And basically, antimicrobials and vaccines have a pretty much controlled epidemic since we invented them, including this one that we are really curbing thanks to our technological developments. But the same very antimicrobial activity that controls infectious diseases is also altering our good microbes, the microbes that have co-evolved with humans throughout always. So this is a double-edged sword. And this effect on our microbiome, we think is driving the trade-off of diseases that, instead of just controlling infectious diseases and being healthy, what we are observing is as societies urbanize, they control infectious diseases at the expense of an increase in immune and metabolic diseases. This is a big challenge. This is what we now have to solve. We know that these immune and metabolic diseases are of early origin, so they have to do with our priming, our programming in early life. And we know that they are modulated by microbes. So there is something called the DOHAD, or Developmental Origins of Health and Disease. And this hypothesis that is gaining more and more support establishes that it's environmental exposure in infancy, during very early gestation and also maturation, that window when we program our modifications in gene expression that alter diseases later in life. And I just want to add that part of that environmental exposure are microbes. We live in a microbial world, and our immune system, the first time when a baby is born or through labor, that is the first encounter with live microbes. Babies during gestation, when they are here, they do have exposure to microbial products of their mothers. This is a very complicated slide, and I left it purposely because I wanted to depict how the choreograph at different lifestyles in pregnancy, in strict lactation, weaning, or in adulthood. There is a very specific choreography of metabolites that are produced by microbes that will later affect disease risk. In some circumstances, they will increase risk. In most non-impacted circumstances, they will protect. So fetuses see microbial products from mom, but it's only at labor that they encounter for the first time live microbes. And this is normal. So microbes have been behind the only two hypotheses before this to explain these new diseases of urban societies that are immune and metabolic. Both hypotheses deal with microbes. The old one is the environment, it has to do with environment, the hygiene hypothesis. They propose that it was reduced exposed to farm animals and to farms and nature in general, to the environmental microbes that increase asthma and allergies. Later, there is a hypothesis called the disappearing microbiota hypothesis that proposes that it is not the environment, but human indigenous microbes that we lack exposure to the right microbes, and this disrupts this maturation of the immune system and metabolic system. And I'm going to show you evidence that both environmental and human microbes, so environmental exposures, environmental microbial exposure, and the microbiome are affected with urbanization. So urbanization, as you can imagine, is a compound of many changes. We change, and this is an example in the Amazon lifestyles and urban and architectural settings in very remote communities in the jungle. And then when they get more exposed, as you move east in the Amazon basin, they get more exposed to materials like you can see zinc and a parabolic antenna, mixing architectural and materials from the previous traditional society. And then you get to towns where you have the houses that we know better. And we ask, what happens in this gradient with the microbes? So we went to South America and got a gradient of urbanization that goes between the border of Peru and Ecuador, jungle villagers. And then as we move east, we found the intermediate, but still pretty traditional rural societies. Then Iquitos, which is a big town, it's the biggest town that is inaccessible by road. You have to fly there or go by boat from Manaus. Manaus is the city, it's a modern city where when you get to Manaus, unlike in the extreme of the traditional peoples, the societies stratify in social classes as we urbanize. So when we got to Manaus, up to Iquitos, lifestyle was pretty similar among all the inhabitants. But when you get to Manaus, you have two cities. You have the poor city and the middle class city. So we had to duplicate our sampling efforts because we thought, these are very different lifestyles. So that was an interesting, we went with the architects and environmental engineers. We studied urban plan changes, architectural changes, air quality, and we went to the houses, and for each functional space, like kitchens, across the gradient, we sample the house, living rooms, etc. And we sample the inhabitants as well as their pets to study microbes. So the preliminary results, and this was the results of a graduate student from University of Puerto Rico, show something that we call principal coordinate analysis. This is a way to show data in a very visual, simple way, where when you have two spheres, each sphere is a sample. When you have two spheres that overlap, that means they are identical in microbial composition in this case. And if they are distant, the more different. So what you observe here is that the red dots, which are swabs from the walls from the jungle town, all clustered together and cluster different in the gradient. If you take a principal coordinate one axis, the next one would be the intermediate society, the village, Puerto Almendras, then Iquitos, and then Manaus, with Iquitos getting very close to Manaus. So there is a gradient in the walls of the houses. There is a gradient in the structure of bacteria in the walls. We call this paper walls talk. And this is due mostly to two factors. As you move from jungle to urban, there is an increased content of human skin bacteria in the houses, and there is reduced soil bacteria. So these are the very houses where people have their babies and babies develop. So their exposure to microbes in the homes change. We also look at metabolites. So we did mass spectrometry to determine what chemicals were there. And again, if we label by colors with red, jungle community, then green, then Iquitos in blue and Manaus in yellow and orange, what we find is a gradient this time on to the left on the x-axis, where you follow the colors with the red, green, and then the blue to the left in this cloud, and then very far in the urban setting, regardless of social class, the metabolites of the city cluster together. Very different exposure of the people that live in those houses to chemicals by urbanization. We observe this is a plot of richness. So as we move in the urban gradient, exposure to different metabolites increases. The diversity of the metabolites to which people are exposed increase. And the reason is that we use a lot of cleaning and personal care products. We invent perfumes, we use medicines, we could see all that, and that adds diversity to our exposures, and that's captured in the walls and floors of the house. Interestingly, we also observe an associated increasing fungal diversity in the houses of Manaus in relation to the houses of lower urban gradient. So we have evidence that urbanization is associated with microbial changes and chemical exposure changes. So what about us? What about the human microbiome? And we know very well there is growing evidence that the gut microbiota, which is our biggest microbial organ in the body, is associated with diseases. Diseases that go from infections or lack of protection to infections to metabolic diseases that are associated with liver diseases, inflammatory, and even brain. So the this is not surprising, but the evidence is really growing fast. So we keep, our approach is more anthropological than clinical. We don't study many diseases. We just want to understand how humans are changing the risk to those diseases with their lifestyle. So we, as a proxy to traditional lifestyles in the human evolution, we study people who are still traditional today. And I study mostly in the Amazon because I've been studying there, almost more than 25 years. And we are interested in, is there anything similar in the cultures of the world that are traditional, or we diverge, geographically? We are different because of geography. Are we different because of geography or because of lifestyle? So we have done viruses studies, studying the viruses that are present in the intestine of Amerindian children versus urban children. And this depicts many viruses. And when we compare Caracas children matching the same age and sex as the children of 3 different villages in the Amazon jungle. The size of the spheres are the abundance of that virus. But what you can see clearly is that the urban site has less spheres, less balls than the other sites. So we have less viruses. We don't know the function of many of these viruses. All these kids were healthy, both in the city and in the jungle. So there seems to be, not surprisingly, I guess, a loss of viral diversity as we move from the jungle to the city. And this work was done with Eric Delwer at the University of California, San Francisco. We also have done studies on bacteria. And what we observe is depicted in this graph. When we look at very remote Amerindian communities in the jungle versus transculturated Amerindian villagers versus rural people in Africa and the US, what we observe is a loss of diversity of bacteria in their microbiome. And this is an association, okay? It's not, I'm not saying that urbanization is causing it, but just this is observational. As we move in the westernization, urbanization gradient, we observe less diverse bacteria in the gut. One of the markers, what increases with urbanization is a group of bacteria called Bacteroides. And there is the disappearance of this purple bacteria called Prevotella. So we can tell which microbes are disappearing and which microbes are rocketing. Very early in a collaboration with Jeff Gordon, we did a study with ages, comparing age of Malawi people, Amerindian, semi-rural people, not very isolated, and the US. And even then, we observe that the first thing is how the microbiota gains diversity. This is a measure of diversity of the microbiota from birth to the 3 years, the first 3 years. It's just rocketing. Kids gain and gain and gain diversity. When they are 3, we cannot tell them apart from their mothers. So before 3, if you give me a swab of the feces of a baby, I can tell you more or less what is the age of that baby. And the second important thing is that you can observe how high the diversity is in the green dots of Amerindians versus the rural Malawians, and here are we in the US. So we can observe the lower diversity there. This is a graph where we took all the data available in data banks in the world by 2017 when we did this study. And we said, let's order the microbiomes of the world by structure, clustering similar ones together and very different ones apart. So this axis called PC1, which explains 22% of the variation, is a measurement of similarity or dissimilarity. The more apart, the more dissimilar. When we put the microbiomes of the world data in this axis by clustering by similarity, what happened was remarkable. And you can see that on the y-axis. So it spontaneously ordered the human microbiomes in the gut by lifestyle, regardless of continent. So the Venezuela, Yanomami that are very remote, clustered next to Burkina Faso in Africa, and then Peru, and then Africa again, Tanzania. Then the second cloud is intermediate. These are all rural in Africa or in South America. And the third cloud on the right are all urban, starting with Japan and China on the left and ending with the Western world. So there is a change in the structure of the human microbiota by lifestyle, more than by geography. Then, because our gradients of urbanization were so wide, we said, well, what about the very early stages of urbanization? When do we start observing any change? Because our gradient in Peru and Brazil was very, very wide. So let's go to the Amazon, and this is where we did the study, right on the border between the Venezuelan Bolivar state and Brazil, right on the edge of the Amazon River basin, which is in yellow here. And there we have different communities. The landing strip community, which we call medium urban exposure. As you can see in the architecture already, they have the traditional hats, beautiful thatch round hats, mixed with houses that use zinc and have windows and a door. This is the medical post, by the way. So they have, you can see the transculturation. They are adopting elements of the mestizo culture of Venezuela, and they are also adopting medicines, versus very remote communities in the same, in the same area that is hard to get to them, and these are photos taken from the helicopter. So these communities, all these border here is an area of river blindness, and there is a program from the WHO to eradicate river blindness. And this is the only spot left in the Americas for this onchocerciasis, is called, for this disease. So we were able to sample these communities, these 5 communities of this type, before they were given ivermectin and after. Whereas the medium exposure has had antiparasites for a long time and antibiotics. So in the low exposure communities, we have data from 2015, pre-parasite treatment, and with the parasite treatment comes doctors who cure infections with antibiotics. So it's really wide exposure to medicine. And post in 2015, 1 year after 3 or 4 visits in some cases of the communities by doctors to provide ivermectin to everyone, an antiparasitic, but then some antibiotics to whoever needed. And what we are observing, and this is still unpublished data, is that the gut microbiota diversity in the low, which we show here, this is a measure of diversity, and these are the age of the people. We observe an increase in diversity with age, which is expected until they are adults. But the green villagers have higher diversity in their feces than the orange villagers who are from the medium exposure. The same is true in the mouth. The green villagers pre-exposed in 2015 had higher oral diversity. but they had lower diversity in their nose bacteria. So as we compare the medicine exposed community, they increased the diversity of bacteria in the nose and in the skin as well. This is, I don't want you to read because it's impossible. I just want you to look at the colors in these heat maps of low villagers, low exposure villagers, and medium, where yellow means high relative abundance of each bacterium, and each row is a bacterium. So in general, I think you agree that the left map is more yellow than the right map, meaning there are loss of relative abundances. So there is we can tell you which bacteria is being reduced. In the case of this bacteria, for example, which is not present in everybody, but in a lot of the low exposure people, it's almost lost in the medium exposure village. So in general, we see loss. The changes are consistent with observed changes from paleofeces. Paleofeces are feces that are found preserved from very ancestral people, a few thousand years old. And they have found that the paleofeces contain high firmicutes, low bacteroidetes, which is consistent with our findings. So when we ask, how are the villagers compared between low urban and medium urban, this time low urban is red and medium or urban is green, how do they compare in the principal coordinate analysis, where overlapping means identical, in 2015? And the answer is they segregate, statistically significant, and this P means highly significant. The blue and the red are apart, both for children and for adults. Now let's move to 2016, after 3 or 4 exposures to medical visits with ivermectin as an antiparasite and other medicines. We lose the differences. So in 1 year, we can see that the community of the very low urban exposure peoples becomes more similar to the community of the medium exposure, which has been visited by doctors, and you know, they have a landing strip for at least the last 25 years. Now, if we say, well, let's take all the Amerindians and see how different are they to Americans, then they are very different. Amerindians are in green, and US people are in orange, and they really segregate apart in both 2015 and 2016. So they are still very, very different to us, but we can see that 1 year of frequent exposure to antiparasites and antibacterials erase the segregation that we observe. We also observe disappearing and gain bacteria in the mouth, so changing the structure in the mouth, with all this bottom part group of bacteria are becoming blue, which means lower, and some of them are gained. In the nose as well, a few bacteria are gained and a lot of them are lost in the nose, although the nasal total diversity increases with urban exposure. And then in the skin, there are a bunch of bacteria that are lost or gained. So with urbanization, there are associated changes in the microbiome of the human body, as well as of the microbial exposures from the environment. That's what is depicted here. Industrialization is associated with lowering the microbiota, increasing diseases, and that's, again, associations. We need to find causation. My former student, Daniela Vargas, also in Venezuela, she looked for a gradient of urbanization closer to a town. So the low exposure here is much higher exposure than the medium exposure in our previous study. But she studied women. The reason being that there is a virus called human papillomavirus that is terrible. Is terrible because it causes the cancer, cervical cancer, which is one of the most deadly cancers in the third world and certainly in the Amazon. The Amazonas state in Venezuela has about 40 times more risk of cervical cancer in women than Caracas has. So she studied vaginal microbiome and human papillomavirus, to which, by the way, we now have a vaccine. And what she observed is that as she moved between the Amerindians from low, medium, and high urban exposure and the mestizos that live in the town, there was a difference in the vaginal microbiome. If she stratified the Amerindians in red into the three groups of low in green, medium in orange, and high exposure Amerindians living in the same town as the mestizos, she could see how the structure of the bacterial communities in the vaginal canal changed towards the mestizo. Why is this important? It is important because it is through the exposure at birth, labor and birth, that the matrilineal transmission, matrilineal means from mother to mother to mother to mother throughout generations. That first exposure of the pioneer bacteria that colonize the body of a baby has been handed off generation after generation on the maternal line. So if you're a boy, you got your first exposure from your mom, but your kids will get their first exposure from their mom, not from you. So it's through mothers, a little bit like mitochondria do. We have the mitochondrial Eve that goes up all the way up to the origin of humans. It also implies that with each generation, if you perturb the microbiota of those moms, they will hand off and pass a perturbed microbiota to the next generation. So we should observe exponential changes in the generation after generations. We care about birth because we think that that seeding is important for the immune system to develop, to mature, recognize self, co-evolve microbes from pathogens. We study C-section babies because they are born sterile. And in this principal coordinate analysis, we can see the mom's microbiota from the mouth in green, from the vagina in red, and from her skin in blue. And then any body site of the baby, including the meconium, if the baby was born vaginally, all the baby's microbiomes cluster similar to the vaginal microbiota of the mother. If the babies are born by C-section, they don't. They instead look like skin. We studied where is the skin coming from that is colonizing the C-section babies. And the answer, we think it's the operating room. We studied operating rooms where C-section babies are born, and the microbiome in the air, in the top of the lamps, of the surgery lamps, and in the walls is mostly human skin, just like what happens in the houses that I just showed you. So it is the built environment microbes that are seeded by humans, and not necessarily the mother in the case of the operating room for C-section, but who knows, the cleaning team, the nurses, doctors. The mother is below, so it's against gravity when they lift the baby up. Those babies are getting bacteria from the human skin first. So why does it matter? Well, we observe that this is age of the babies, and this is the position of the structure of their gut or fecal microbes. Babies born by C-section remain different in their development of the microbiota. They do mature the microbiota, but in a different way than normal babies born normally, vaginally. And we know of another association, and is that C-section babies grow bigger. This is what we call CETA score body mass index of babies from birth to the first year of life. In orange, these are babies born by C-section, and in green, babies born normally. These babies tend to have an accelerated growth if they were born by C-section. So this is intriguing because growing too fast is a risk factor for metabolic diseases later in life. But what we don't know is causation. What I've shown you so far is all associations. Associations, this is what we observe. How do we study causation? Well, a great way is to use animals where we can let them have their babies vaginally, or we can do C-sections, in which case we have to foster the animals to another mom because we haven't been able to do survival C-sections. And then we can restore and try to understand which microbes normalize if there is an effect on development. So this takes a village of people. These are members from my lab, postdoc and associate researchers, 3 different labs of experts at Rutgers with 3 different expertise, and then a bunch of students, graduates and undergraduate. And we are a big team because we study a lot of things. We are studying 10 families that are born vaginally or by C-section, restored or not, with microbes. And we look at early age, gene expression, inflammation in the colon, hormones, cytokines in the blood. We study body weight and food intake. We study the fecal microbiota at different points. We study the brain and behavior and bone phenotype early and late, and markers of metabolic disease. Because we hypothesize they would grow faster, just like humans do. So the first results I'm showing here are the C-section red dots, microbiome in the feces of the mice. They cluster apart from the blue feces from mice born vaginally. And what we observe is that these differences, if we do microbiota development, if we plot how the microbiota develops with the age of the mice, then we get a curve because the microbiota is maturing. If we plot the C-section mice against the same plot, what we observe is a flat line where the mice born by C-section seem to have a premature microbiota. They look too old for their age, or an immature microbiota later. They look too young for their age. They have a microbiota of 4 weeks old when they are 6. So this is telling us something that is causal. This is caused by the treatment, by the intervention. We study intestinal gene expression, and the first experiment, we wanted to get it very early, because what happens when you have a overweight mouse is that the overweight itself is changing the microbiota. So we want to go very early, before the disease appears, before the phenotype appears, to be able to predict what was different. And what we can tell, looking at 1-week-old baby mice that were born vaginally, they have this pattern of colors in the gene expression where green is high and red is low. And C-section has very altered pattern of gene expression. And this is the CS here. They have red where there should be green, and vice versa. Restoration does not normalize much at 1 week. Now we are studying at later weeks. And then this is the vaginal animals that are fostered. And as you can see, there are some irregular genes here. meaning maybe just the act of fostering is stressing the animals and changing the gene expression. That's another, it opens another whole new path of research. Animals grow bigger if they were born by C-section versus vaginal, much bigger, especially females. And when we restore on the right, we see the weight of the vaginally born or foster, C-section, restore, and C-section. That's the order. So restoration partially restores, partially normalized, but not entirely. And the phenotype is stronger in females. So now we have something to work on, which are the bacteria that are protecting. And one of the problems we observe is that C-section born, some of them are overweight, not 100%, 56% are overweight. If we restore with exposing to maternal vaginal microbes, only 12% are overweight. So there are responders and non-responders. We are very interested in the non-responders. What do they have that are protecting them? We measure intake of oxygen, so metabolic rate, and what we observe is that in relation to the pink vaginally born, the C-section females, C-section born females have lower metabolic weight at night. And that is consistent with gaining more weight because they are spending less energy. Restoration seems to partially restore. So in summary, C-section causes gain in higher gain in body weight, lower metabolic rate at night, and microbial restoration partially normalizes. We are still studying the phenotypes. So what about in humans? Do microbiome restoration help? We did vaginal seeding in humans, in human babies born by C-section, by exposing them to a gauze that was incubated in the maternal birth canal. When the baby is taken out, we pass that gauze first through the mouth, then the rest of the body. We published that in a short paper as a pilot because we observed that we could partially normalize the microbiome. Recently, there is a study where they did fecal transplant in newborns, also trying to see if they could normalize the C-section born. And this is a short video of our first baby of the study being born by C-section. Once they dry the baby, cut the umbilicus, take their baby to the station, that's the vaginal gauze that we pass in the face and in the rest of the body. So we did that in a US, Spain, Chile, and a few Bolivian babies, 174 families, 177 babies, of which 28 were seeded after C-section, 49 were not. And we have almost 100 vaginal births to compare what is normal. And these are the results. The blue babies are the microbiota maturation with age. The C-section babies are abnormal, as we saw in mice. And the green line shows the C-section restore, which are a little closer, partial restoration in the feces, good restoration in the mouth, and good restoration in the skin, where the C-section has the biggest impact. So if the babies are born naturally, they have a pattern of trajectory of maturation. If they are born by C-section, their pattern of microbiome development is abnormal. But if after C-section you give natural microbes, you can partially normalize. So the practice became viral, and now a lot of mothers are requesting it. This is not a normal medical practice. We need to be aware of risk of infection. So our moms were highly examined, thoroughly examined before this procedure. And we need to understand which are the microbes. And to understand whether this really protects from having the impacts increasing diseases, we need randomized controlled clinical trials, because and this is urgent because a lot of women are requesting vaginal seeding, and because we need to prove safety. So there are 3 clinical trials that are currently being developed. Ours, one in Mount Sinai, and one in Switzerland, in Stockholm, in the Karolinska Institute. So why the real stuff? Why vaginal seeding? Well, like for blood transfusion, we haven't been able to create synthetic blood yet. And the same with fecal transplant. There is nothing like the real poop to restore people who have Clostridium difficile infection. People are working trying to create the synthetic mixes of natural bacteria to restore and protect. We are not there yet. Same with the vaginal seeding. So if industrialization is degrading the microbiome, and that is associated with increased disease, can we restore and stop the trend or revert it? For that, since I've shown you that we are losing microbial diversity, we need banking of microbes right away from the microbiota diversity of the world. So we created the initiative of the Microbiota Vault with the idea of having long-term preservation of diversity. This long-term preservation comes from local collections, which are the live collections where research can be done with global cooperation, education, technology transfer to do the research that is needed to stop the current trend in diseases. You can look at the Microbiota Vault in the webpage. We have an amazing team of scientists and 2 Nobel Prizes. Just have a look. And with that, I thank everybody for your attention. After thanking all the many labs and collaborators that have helped us understand a little bit better where we are with the impacts of the microbiome and urbanization. With that, I'm happy to take questions if you have them.
Thank you so much, Gloria. That was great. We do have some questions. Do you have any data regarding how diet ameliorates the urban microbiome phenomenon?
Yes, so not from our lab, but so for babies, breastfeeding is highly restorative. So even babies that are born by C-section, because the milk contains prebiotics or substances that encourage the growth of good human bacteria, breastfeeding is a very important diet that should be respected at all costs. Later in life, we know that high fiber is super important. That again, it's important not to nourish yourself, but to nourish your microbes. And your microbes in the hindgut, in your intestine, are nourished from the parts of your diet that you can't digest. And that is pretty much resistant starches and cell wall polymers of the plants. So a high vegetable diet is extremely important. In the Amazon, people eat about 100 grams of fiber a day. And if you read our standards and recommendation, we think 30 grams is high. It's not high. We should be eating a lot more than that. Oh, and of course, a lot of fruits and natural foods, non-processed. We have an illusion that with urbanization, our dietary diversity increases. And it's an illusion, because if you are eating pasta and then a cheesecake and then a cake, these are basically reduced to the same thing. It's the extracted, simplified ingredients of sugary carbohydrate components. When you eat a natural fruit, you are eating a lot of antioxidants and vitamins, and the diversity of metabolites of the diet is much higher if you don't process. Unfortunately, so what we have done is just market what humans crave for. We crave for fat and we crave for sugars because these were very non-abundant during human evolution. Our ancestors didn't have access to fat and sugars. So we developed a crave that is evolutionarily wired. And now we have markets that exploit that, and we love, who doesn't love potatoes, fried potatoes. We love oil, fried stuff, and high fat, and sugars. So again, we need to understand this and be able to modify our behavior and control. I don't say don't eat ice cream, but just control it and don't eat it every day, and eat high fiber, which is the single most important component of a provision for a healthy microbiome.
Great. Other than diet, are there any specific actions that we can adhere to improve our microbiome?
Well, certainly, when, if it's natural, it has to be better, because that's what we evolved to. We are a product of our evolution. So we have to understand what are we changing that is unnatural. So when mothers ask me that question, I always say, do what is natural. So hug your baby, skin to skin. At home, just allow your skin to touch the baby's skin. Be with the baby the most possible and breastfeed, and kiss your baby. All with technology, what we have done is put the babies away from the mothers. We use technology. We leave the babies in rooms that are empty, full of skin microbes, as we know in the urban houses. And then we monitor them with our technology. Then we use technology to carry them as well. So we have impaired the necessary bonding and direct contact with the babies. I think that's something we need to gain awareness of. Again, natural diets and processed diets are extremely important. Reducing unnecessary use of antibiotics. This sounds like, obvious, but don't think it's that obvious. You can buy antibiotics over the counter together with the batteries in third world countries, the whole world. Many people think that just preventive antibiotics are a good idea. They are not, especially for children. Reduce C-sections when they are not necessary. C-section and antibiotics can save lives, and thanks God we have them, but you have to use them really in a responsible way. C-sections are being used by choice in countries like Brazil, especially in the cities. Rio de Janeiro, C-section rates is over 90%. 90. So nobody's giving birth naturally anymore. In part, what is to blame? Well, we should be doing more research on how to give birth naturally with less pain, instead of taking shortcuts. So again, this is a call to learn to be technological and more natural.
Okay. Do you have any data on populations that consume raw milk versus pasteurized milk? There is a trend internationally to return to consumption of raw milk in first world nations because of gut microbiome concerns.
Yeah, I don't, but I know of that trend. And again, fermented foods. So the thing to worry about are infections. So that milk, it would be a cow milk. And most of our infections that are digestive infections are zoonotic. They come from the animals we have in agriculture, in animal production. And that is remarkable. When you go to the jungle and you drink the water that somebody has in the village, where all the kids are putting their hands full of feces, you don't get sick. We never get diarrhea, because most of those pathogens come from cows and chicken. So the concern is always, is there a zoonosis risk? But again, fermentation is one way that was used from a long time ago to preserve food, so the food doesn't spoil. And I think we are, again, going back to more natural ways of preserving food and being exposed to fermentation products, which are known to be healthy. And we are starting our return But we have to remember that we are starting our return in the places where we first started with the impacts. So it will take generations probably for the whole world to go back. Meanwhile, you know, generations of children will have a compromised health.
Okay. Do you see any significant health issues arising soon due to the extensive use of hand sanitizer and the lack of human interaction during the pandemic?
Yeah, so we wrote a perspective about that. The use of sanitizers is, of course, highly justified now. And we discussed the possibility that that will be changing the microbiome of people, you know, again, impacting microbes. On the other hand, the children have been in their houses and much more in contact with their parents than we were before the pandemic. So to what extent we may be compensating, the children are not, you know, exposed to antimicrobials and alone. They are more with their siblings and with their parents now. So the answer is we don't know, but one thing may be compensating the other. We didn't isolate, or not everybody isolated entirely from other human beings. I know some people did, people living alone in Manhattan. I know some who just lost contact with other human beings, and that's terrible. Not only from the microbial point of view, but also human, you know, we all need to socialize. Thanks God we have this technology that allows me to be with all of you today. We will see what the effects are. I know some people who are following the pandemic and trying to understand if there are effects.
Okay. I think we have time for one final question. How effective are over-the-counter probiotic supplements in counteracting the effects of antibiotics?
That's another very good question. The answer is we don't know, because they haven't been properly studied. So we have the problem that because they are natural and you put them in food, then you can sell them. But if you do claims of that put the product into the pharmacy, then they lose the market. You can't say that this is anti-inflammatory, because then the FDA will require a prescription. So we are in a, you know, in a situation where basically there is not enough transparency. I'm sure companies have data, and we microbiome people have the impression that most probiotics don't do anything bad or good. And we need to place that research outside the private companies. It has to be in the, you know, public sector or universities, where we start to study the real human probiotics and have strong data. And if it's anti-inflammatory, then we will deal with the FDA. The FDA requested to me to have a meeting because they consider our exposure to vaginal microbes of C-section born a new drug, because our hypothesis is that we were normalizing microbes that are important for health. So we had to have a permission from the FDA and IND to do the study, which we did. But that's how, you know, if you're a company and you're selling over the counter, you don't want to get your product to a pharmacy and have prescription. So I think there is a conflict there between the research and the interest.
Okay. Well, thank you so much, Dr. Dominguez-Bello. It's a great presentation. I want to thank everybody for joining us this afternoon. We will have the recording posted, and we will send it to registrants and attendees following this event. I wish you all a very good day, and have a great summer if we don't see you before that.
Thank you. Thank you, everybody.